Today we will do the experiment about preparation of mitophase chromosomes from culturing cells. First, I will introduce what is a chromosome. Can you see the picture here in the eukaryotic nucleus? This structure, thread-like structures. Can you see? Here is a thread-like structure and consisting of chromatin, and this carry genetic information arranged in a layer sequence. We call this structure chromosome. And today we want to get this. And the structure of chromosome, we can see each chromosome has two arms. One is short and another is longer. The top section is called P arm. Because it is small, so we call it P. And the bottom section is called Q arm. Because Q just in the alphabet is followed P. It didn't have any other meaning. It's just followed the P. So we use Q to stand the long arm. And the structure that separates the two arms is called centromere. Here is the centromere. This is the whole structure of a chromosome. And for most of life of cell, chromosomes are too elongated and tenuous to be seen under microscope, just like this. It's very, very long, and we cannot see the clear structure here. However, fortunately, before a cell gets ready to divide by mitosis, each chromosome is duplicated. And the mitosis begins the duplicated chromosome condensed into short structure. So you can see in the mitophase, the chromosome become more condensed, so can be stained and easily observed under the light microscope. Here we can see it. Just use a microscope. And in this mitophase, the chromosomes separated from each other. So if we get the chromosome in this stage, then we can get the separated chromosome and very clearly structure. So we want to stop the mitosis in the mitophase. And that means we do not want the cell divide. We just want the cells stay here. And how we do this? This is an agent, calcimide. This can depolymerize microtube and limits microtube formation. That means it can inactivate spindle fiber formation. From this picture, we can see this is mitophase spindle, and this agent calcimide can stop the spindle formation here, and the spindle will pull apart the chromosome. If the spindle is inhibited, so we just leave the chromosome here in the cell. And then we can arresting the cell, or we can harvest the cell in metaphase, and allowing cell harvest and karyotyping to be performed. So today we will stop the cell here, and we want to get the chromosomes. And the objective of today is just to prepare the mitophase chromosomes from culturing cell. This is our objective. And the material and the instrument for today's experiment First is the subculture cell, HeLa. Everyone are very familiar with this cell. And the flask sterile peptide culture medium solution is 10% fetal bovine serum DMDM. And the microscope PBS, and PBS we used lots of times. 0 to 5% trypsin solution, and this is a new solution we first use hypertonic solution. 0.075 micromole per liter potassium chloride, and fixative. This should be fresh made. So this is methanol and acetic acid. The ratio is 3 to 1, and the ratio is volume to volume. This is the fixative. And this is our important agent today is Cosimid 5 microgram per milliliter. This is all the material and the instrument. And the protocol for today is first we should culture the cells for about 2 days with DMEM. Then I give each group 1 flask. The cell is on the bottom, lots of cells on the bottom. And before our class, 4 hours, we add Cosimid. The final concentration is 0.07 microgram per milliliter, and mix well. Incubate for 4 hours and 37 centigrade before harvesting. So what we will do first is harvest cells and centrifuge. First I will give each group 1 flask, and in the flask there are HeLa cells. We are very familiar with HeLa cell, and what we should do just remove the culture medium from the flask, and just use the pipette and remove the culture medium into the centrifuge tube. After that, we can add some PBS in the flask. Just cover the bottom, then gently shake the flask. Then you can discuss the PBS. Just like first, this is the flask, and remove the medium into the centrifuge tube. This is the medium, and add some PBS, then discard the PBS. After this, we just add the trypsin into the flask. Can you still remember the experiment of subculture? We just do this just like the subculture, and we pipette 1 milliliter trypsin in the flask and incubate for 2 minutes and 30 seconds. However, during this step, there are a little different between this and the experiment of subculture, because in the subculture we discard the trypsin first, and then we stand for another 40 minutes. However, in this step, we just add the trypsin in the flask, and we didn't discard the trypsin. We just add the culture medium back to the flask. And can you understand here? Okay. Then we will produce our cell suspension, just mix by pipette up and down, just like you did the subculture, and then put the cell suspension into the centrifuge tube and centrifuge and 1050 RPM for 10 minutes. there are something I should remind here is this is our centrifuge tube. However, this is another tube. Before we put the tube into our centrifuge, we should put the centrifuge into another tube, just like this. However, first we should make sure two of the tubes are balanced. So you just put all of this into the balance. Balance is on the table. Two of this. If they are not balanced, what should we do? We just use a pipette, add some water. Do not add the water into your centrifuge tube. Just add the water to the outer tube. Understand here? Then after the two tube balanced, we put this into the centrifuge and the centrifuge for 10 minutes at the speed 1,500 RPM. Then after this, we will remove the supplement completely. After the centrifuge, you can see your cells in the tube. Maybe this side, maybe another side. However, I'm sure you can see it. Then, if you can see the cell, you just pull the medium from the opposite side. Can you understand here? If the cell is here, you just put like this. Because if you put from the cell side, it will discard the cell. So gently remove the supplement, and only once just remove the supplement. Understand here? Okay. Then we just harvest the cells. After this, we will add 8 milliliter of 0.075 micromole per liter potassium chloride here, and pipette up and down. Here we need so many times, 60 to 70 times, just upside down, in order to make sure all the cells mixture. And after this, we will incubate the tube for 20 minutes at 37 centigrade in the water bath. Here we can see there we need to wait for 20 minutes, and during this time I will give the lecture. Understand here? Okay, let's start our experiment first. PBS is here. And then we add 8 ml of 0.075 micromole potassium chloride. Its time is for you. Okay. Because the time for each group is different, so I will finish here, and you will do next step. Then I will continue to say. After incubate for 20 minutes at the 37 centigrade in water bath, just add 1 ml fresh prepared fixative mix well, and then centrifuge the cells again for 10 minutes and remove the supplement. So I will give some group the fixative.
30 seconds. You could just 30.
There's more.
There is.
You didn't incubate it.
Just enough to.
There is.
So we know this is done. Like, that is
It doesn't
right?
we added 8 ml of 0.075 potassium chloride, pipette up and down for 60 and 70 times. For this step, because this is hypertrophic solution, so in this solution, the water will go into the cell. The cell will swell. So the time for incubate is very important because the incubate time is too long. Then the cell will swell and will break. So we cannot get the chromosome. However, if the time is too short, we cannot get a very separated chromosome. That means the chromosome will not separate very well. So the time for this step is very important. And this solution just makes the water go into the cell. And the step 6, we add 1 microliter fresh prepared fixative and mix well. For this step, because after the step 5, if we centrifuge, the cell will break. And so we just add the fixative. This fixative will keep the shape of the cells. Will keep the shape of the cells, and when we centrifuge, the cell won't be broken. And then we centrifuge the cells and remove the supernatant. We will get the cell. This is step 7. And then we resuspended the cell and fix the cell by adding 8 microliter of fixative incubator in 15 minutes at room temperature. This is the real fixative because last step we just add 1 ml fixative, and this time we add 8 ml fixative, and we need to wait for 15 minutes. That means this will give the cell and the chromosome very well shape, and we will observe it clearly in the following step. This is the step 8. And step 9, after 15 minutes and room temperature, we will centrifuge again for 10 minutes and remove the supernatant. Here we will get the cells. After the last centrifuge, we resuspend the cell in a small amount of fixative, 3 or 4 drops. You know, after the last steps, you know, when you discard the medium, even you discard the medium completely, there are still some medium live in the centrifuge tube. So I will help you to observe the solution, and I will tell each group how many drops you will add in the tube. I will help you to do this. Okay, understand? Here is step 8. Everyone is clear here? Okay, and then we will hold the pipette. This is sometime I need the slide. This slide, I will just show. Just like this slide. This slide is different from our normal use slide. It's cold. It's very cold and cleaned. We hold the pipette 15 centimeter. That means it will high from the above the slide and drop the suspension onto the side. Understand here? And after you drop the suspension, you can see this is a slide. You drop 1, 2, 3, drop 2 or 3 drops onto the slide. Then you just like this, blow across the slide for 2 or 3 seconds. That means just blow 1, 2, 3 times. It's okay. And this will help spread the chromosome from the ruptured cell. Sometimes I just said if the hypertonate time is too long, our cell will break and we will lose our chromosome. However, in this step, we want our cells break. Can you understand here? Because this is a normal cell, and this is our swelled cell. There are some chromosomes. However, the chromosome is still in the nucleus. We just want at this step the cell break, and we can see the chromosome. Can you understand here? However, we should keep the cell completely here, just break here, not early. Okay, this is the step 12, and step 7, we will drive in with the fire. Fire is on the table. We will drive in with the fire, and you should remember which side the suspension is on, because next experiment we will stain the chromosome. If you forgot, so you should remember. And then we will dry for 7 days, and next week we will do the gymnast binding, another experiment. So today, so next week you needn't hand in your lab report, because this you can write today's experiment and next experiment together and hand in the 3rd time. Okay, this is all our protocol of experiment. And note here is I just told you the hypertonic treatment causes a swelling of the cell, so the optimal time of treatment varies different cell types and must be determined empirically. That means too long or too short is not okay. You should vary exactly. And if the sides are not made the same day as the harvest, fill the tube with fresh prepared fixed tube. Tighten the cap and store the suspension in the refrigerator. Here that means, however, we will do this today. If you have some suspension, you just want to do the experiment tomorrow, you just put it in the refrigerator. That doesn't influence the result. This is all the experiment today, and we will continue our experiment.